What you are about to see is a presentation that I did for my astrobiology class in fall quarter of the year 2020, in which we were supposed to choose a body in the solar system to send a mission to search for evidence of extraterrestrial life. And I chose the dwarf planet Ceres in the asteroid belt, not because I wanted to look for presently existing life necessarily, but because I thought it might shed some light on how our form of life came to be, on the origin of life on Earth. And so that's where we're going to start. We're going to start with the primordial Earth, which formed about 4.55 billion years ago. The moon formed around 4.5 billion years ago. The Earth cooled sometime between 4.4 and 4.0 billion years ago. So that's around the time the Earth would have first been able to support life as we know it. And the earliest evidence we have for life on Earth is from about 4.28, almost 4.3 billion years ago. So possibly even earlier than we think the Earth might have been cool enough to support life by. Um, and that's really interesting because the time frame between when Earth first began to cool enough to support life and when life first appeared is incredibly short. And that has caused some people to question whether life evolved here or whether this planet was simply colonized by already existing life. And of course, that brings us to panspermia. Now, there are two major two major types of panspermia to consider when we're talking about how life ends up on planets. The first is lithopanspermia, which is panspermia by rocks, basically. Um uh, a meteor or something hits a planet, and impact ejecta is kicked back up into space, and sometime later on, that ends up falling down on a planet, and that planet gets colonized. The other type of panspermia we need to consider is molecular panspermia, the idea that organic molecules, um, such as we would need to form the first life, are pretty much present already in space, and so they fall to planets and become true life forms. When we're talking about lithopanspermia, um, a sort of proof of concept of that is the fact that there are microorganisms on Earth who can survive space-like extremes. They can survive low pressure, low temperature, low moisture, and high radioactivity. We also have microorganisms on Earth that can survive extreme acceleration and can survive extreme deceleration, like sudden impact. So we know that there are life forms on Earth who could survive conditions such as would be necessary in order for organisms to make a trip through space from one planet to another. And here we need to take a brief diversion to talk about mitochondria and macroorganisms, because although there are many microorganisms who can survive space-like conditions, including ejection into space and reentry, um, they're all prokaryotic, like bacteria and archaea. Uh, in order to get macroorganisms, things like fungi, animals, plants, you need mitochondria. Really, not all multicellular organisms are eukaryotic, but all macroorganisms are. So all the multicellular organisms that are big enough to see and that are fairly complex with a lot of different cell types, those are eukaryotes. Those aren't prokaryotes. And all eukaryotes that we know of either have mitochondria or their ancestors did. Um, so it appears that mitochondria um, and eukaryotic cell structure in general are necessary for what we consider complex life or complex, obligately multicellular life. Especially the mitochondria, because that's where we get most of our energy from. Eukaryotic cells, thanks to their mitochondria, are capable of producing a lot more energy than prokaryotic cells are. But where did our form of life, where did eukaryotes come from? Well, it's been thought for a long time that we eukaryotes are more closely related to the archaea than we are to the bacteria, number one. But the mitochondria in our cells seem to be more closely related to the bacteria. The mitochondria in our cells, they have their own DNA, and that DNA is more similar to bacteria than archaea. Meanwhile, our nuclear DNA, the DNA that makes us what we are in the nucleus of our cells, that DNA seems to be more closely related to the archaea. And so the archaea and the eukarya are generally considered to be sister groups, and the bacteria much more distantly related. Although um, it's been looking more and more over the decades that the eukaryotes actually evolved from within the archaea. We aren't sister groups. They're not more closely related to us than the bacteria. We actually evolved directly out of them. We are pretty much a type of archaea that that took up a bacterial endosymbiont into our cells. Specifically, it looks like the mitochondria evolved from rickettsial endosymbionts, similar to the bacteria that causes rickets, and the host cell DNA, the DNA in our nuclei, seem to be most closely related to the Asgard archaea, or the this image here shows the Heimdall archaeota. I've seen studies that also put us as more closely related to the Loki archaeota, but definitely from within the Asgard group, or the Asgard archaeota. Also known as the eukaryomorpha, fittingly enough. So how did this happen? How did we eukaryotes end up with a cell in which the nuclear DNA that makes us who we are seems to be evolved from within the archaea, but yet the mitochondria, which basically function as little cellular batteries, giving us the energy to have complex multicellular life, that seems more closely related to the bacteria. And there are a lot of different hypotheses on how this eukaryogenesis happened. And it usually involves some kind of symbiosis. Symbiogenic theory is technically the hypothesis, or well, I guess it's a theory now. It's a full-fledged theory. It's not just a hypothesis anymore that the mitochondria in our cells evolved from bacteria. The first hypothesis for how this happened exactly is the Archaeozoan hypothesis, and that basically states that our cells were already proto-eukaryotes before taking up the mitochondria. So first they developed a nuclear envelope, or a nucleus, and other eukaryotic traits like phagocytosis, long before we took the proteobacterial endosymbiont into our cells. That's contrasted with the symbiogenesis first hypothesis, which states that the original eukaryotic cell, or the ancestor of the eukaryotic cell, was basically a typical archaean, and we did not start to develop our eukaryotic traits until after the mitochondria was acquired. And that the mitochondrial acquisition, or mitochondrigeny, is what triggered the nuclear cytosol compartmentalization that characterizes eukaryotic life. Of course, there's also the fat hypothesis, or the phagocytosing archaean theory, and that states that the host cell was basically like a typical archaean, basically prokaryotic, except for one difference, which that it was already capable of phagocytosis. And that's how it acquired the archaeon, was it tried to consume an alpha proteobacterial endosymbiont. There's also the serial symbiogenesis hypothesis, um, which seeks to explain why the outer membrane of the eukaryotic cell looks a little bit more like a bacterial membrane than like an archaeal membrane. So basically, we absorbed an alpha proteobacterial endosymbiont, but then a bacterium tried to consume that, or maybe the bacterium tried to consume an archaeon first, and then that conglomeration of creatures tried to consume an alpha proteobacterium. So we don't really know the order things happened in, although I personally am a proponent of the symbiogenesis first hypothesis, that the mitochondrial acquisition, or the mitochondrigeny, is actually the event that triggered the nuclear cytosol compartmentalization and allowed us to subsequently acquire our eukaryotic traits. So back to extremotolerance and microorganisms, what's really interesting is that most of the organisms that could have hypothetically survived a trip through space, say from Mars to Earth, um, are bacteria. It's mostly bacteria who can survive low pressure, low temperature, low moisture, high radioactivity, high acceleration, and extreme deceleration. It's mainly bacteria who are famous for being able to survive those kinds of extremes, whereas the prokaryotic organisms that we eukaryotes, or at least the original host cell with our nuclear DNA, evolved from, the archaea, they are much more famous for being able to survive high heat and pressure, high acidity, high salinity, high alkalinity, um, more planetary extremes, more Earth-like extremes. And so that would at first appear to form a kind of contradiction. I mean, the prokaryotic organisms that we're most closely related to don't seem to be the ones that could have survived a trip through space. They seem to be the ones that are better at tolerating much more planetary extremes. So, is this a paradox? Is it a contradiction? Or could there have been two origins of life? One on Earth producing the archaea, and one on Mars, for example, producing the bacteria. Could the bacteria look like they are better at surviving a trip through space because the bacteria we have on this planet are descended from organisms who survived a trip through space? Could there have been two independent origins of true cellular life from one common genetic ancestor? And to answer that question, we need to look at organic chemistry in space. So, elements and compounds that are necessary for life, or which would have been necessary for abiogenesis, which can be found outside of planets, those include, carbon, oxygen, nitrogen, sulfur, phosphorus, all the basic elements you need to make most of the organic compounds in our cells. We have also found evidence of amphiphiles in meteorites, sugars, including ribose, which was a fairly recent discovery. Um, we have found amino acids in the Murchison meteorite, and I believe even recently we've found the first protein in space. In addition to that, we have found evidence of nucleobases in meteors, including the purines, adenine, and guanine, as well as the pyrimidine compounds uracil and cytosine, although cytosine has never been confirmed at high enough above background levels to say for sure that we actually found it in the meteor itself and that it wasn't a contaminant from Earth. And from that, we can kind of posit a rough timeline for how life would have evolved in space. At first, we would have amino acids and purine and pyrimidine in interstellar gas clouds. Then the purine and pyrimidine pardon me, nucleobases could have formed in interstellar ices. There have been experiments showing that if you use UV radiation on purine and pyrimidine, you can get the four canonical nucleobases that are found in our ribonucleic acid in interstellar ices, or at least in experiments using interstellar ice analogs. Uh, Sugars were probably present in the protoplanetary disk. Like I said a moment ago, we found evidence of ribose in meteorites. Peptide nucleic acid could have formed due to high-speed collisions between grains. Um, we know because of experiments by Jennifer Blank that peptides can form when amino acids are hyperaccelerated and then suddenly stopped. Um, we know that from ballistic experiments, the ballistic experiments that Jennifer Blank performed. Um, and it may even be possible that nucleosides and phosphorylated ribose and nucleotides formed around this time period as well. And that could have definitely given us peptide nucleic acid, which is a hypothesized precursor to ribonucleic acid. Then the sublimation and condensation cycles of spinning grains along the H2O ice line of the protoplanetary disk, um, that may have allowed these RNA molecules or PNA molecules to base pair and hydrolyze. And that could have been the first form of replication, was cycles of base pairing and hydrolysis. And then we have found amphiphiles and liposome-like structures. Well, we found amphiphiles which could form liposome-like structures in meteorites, and those could have possibly formed liposomes in the subsurface water of planetesimals, where phospholipase backbones would form in montmorillonite clay sheets. And that brings us to Project Pepsi, the protoplanetary evidence of prebiotic synthesis investigation. And we are going to look to the asteroid belt. The closest analog we have for the protoplanetary solar disk is generally assumed to be similar in composition to the protoplanetary disk, although it's thinner. The interplanetary, or I guess in this case, the interasteroidal space, the space between the asteroids, is pretty much a vacuum nowadays. It's just like interplanetary space, whereas the protoplanetary disk would have been a little bit thicker than that. Not much, though. This is actually, the asteroid belt is probably an okay-ish analog for the protoplanetary disk. And we are going to look specifically at the dwarf planet Ceres. Why? Because it's a lot like we would expect from planetesimals in the later stages of the planetary coalescence. If we look at Ceres, it has a carbon content 7 times greater than most carbonaceous asteroids. At one time, it probably had a subsurface ocean. It may still have a subsurface ocean today, although it would be really briny. It would be extremely high in salt. And Ceres has montmorillonite clays, the type of clays that are known to be conducive to the formation of phosphoribose backbones. There's also cryovolcanism on the dwarf planet, and it has a constantly changing surface. So this is an active world where things are constantly churning around. And so from here, we're going to go to Mission Caspia, the Sarian Astropalaeontological Search for Protoplanetary Evidence of Abiogenesis, part of the Protoplanetary Evidence of Prebiotic Synthesis Investigation, aka Project Pepsi. And the first part of Mission Caspia would be to send the orbiter Lucretius or Lucretius if you prefer, to the dwarf planet Ceres. Now the Lucretius orbiter is named after the Roman poet who first described evolution via natural selection. It will be equipped with a remote Raman laser spectroscope. It will have a visual and infrared spectrometer, and it will deploy the Pliny lander, named after Pliny the Elder, creator of the world's first encyclopedia and an unstoppable beast of a scientist in the ancient world who died studying the eruption of Mount Vesuvius in Pompeii. I'm not going to put any spectral analysis equipment on it, but it will collect a soil sample to bring back to Earth. And of course, the Pliny lander will deploy the T.H. Huxley rover, named after Darwin's bulldog. This little guy will be equipped with an evolved analyzer, or an evolved gas analyzer, quadrupole mass analyzer, EGA QMS. It will be equipped with a matrix-assisted laser desorption ionization time of flight mass spectrometer, aka MALDI-TOF, which is really good at analyzing organic molecules, especially big complex ones. And it will also be equipped with the search for extraterrestrial genomes gene analyzer. In case we find anything on the planet that actually has RNA, we can analyze it and maybe even see what it's most closely related to. And that brings us to the end. Stop. Actually, there is one thing I want to say at the end here, which is that when I first came up with this hypothesis that life evolved both on Earth and on another planet, for instance Mars, and that it came from proto-life forms in the later stages of the protoplanetary disk, I was working under the assumption that there wouldn't have been a lot of water in the protoplanetary disk and that cells would have evolved later to deal with the watery conditions on ancient Earth and ancient Mars. And that hypothesis did have a little bit of a failing, which is that the 355 genes shared by all life forms on Earth, those genes include some membrane encoding genes, which makes it seem like the common ancestor of the archaea and the bacteria probably wasn't acellular. It probably did in fact have cells. And that was a failing of my hypothesis, except now we're getting to learn that water is a lot more common in the solar system than we used to think. Not only are there several ice moons that are thought to have water on them, but Ceres, it looks like, might have a subsurface ocean, and it's looking increasingly likely that that the planetesimals in the later stages of development in the protoplanetary disk probably had subsurface water. So the idea that there were two separate origins of true life from a genetic common ancestor is kind of looking less likely. It appears as though the common ancestor of the archaea and the bacteria did have some kind of a cell membrane, but that doesn't negate the possibility that that was an adaptation to watery environments, because now we know that the protoplanetary disk probably had significantly more water than I was originally thinking. So the idea that the archaea and the bacteria share a common ancestor in the protoplanetary disk before the formation of the planets is still a valid idea. It's just that now it seems like that common ancestor was in fact a true life form. It was not a proto-life form, and so there probably weren't two separate origins of true life. There was probably just the one, but yeah, it may have occurred significantly before the formation, or before the not before the formation of the planets, but before the modern planets had finished forming, back when they were still just planetesimals, or even earlier than that. So yeah, that was my astropaleontological presentation, and I hope you enjoyed it. Peace out.